move. Okay, it's well, unfortunate not to meet in person, of course. I'll start to apologize because due to budgetary restrictions, I'm now in a corridor, so there's no door really to my office. So I hope I won't be interrupted too much. Let me share my slides. Okay, here we go. So I'm going to talk about the use of approximate Bayesian computation in phylodynamics, and I'll try to define all of these terms. First, I'd like to thank the people who actually did the work, especially two brilliant PhD students, Gonche Danesh, who's currently in her 4th year, and Emma Saulnier, who's now a data scientist. And this is also in collaboration with Marc Choisy, who's based in Ho Chi Minh City now, and also Olivier Gasquel, who was in Montpellier but is now in Pasteur. Before beginning, I just wanted also to talk a little bit about context. We talked about the pandemics and this crisis, but in France, we're also struggling right now in terms of research. Here I'm showing the positions at the CNRS and the trend over the last 20 years. And well, unfortunately, we're talking about data. This is a really good correlation, which is very depressive. We've been losing 10 positions a year, and this year is no exception. And more globally, for those who saw it in Nature even, we've been struggling right now with a law that is about to destroy whatever was left of the French independence in research. In fact, I'm sorry this slide is in French, but when you look in terms of budget, the government is putting forward a huge budget increase, but when you look at the numbers, it's actually a decrease compared to what we've had over the last years. But even more importantly, and you can have a look at this conclusion of the CNRS Ethics Committee about what we're living right now. And this is really a huge danger in terms of the precariousness of the research personnel, in terms of putting competition as the main motivator, which will obviously lead to misscience, as we call it, falsification of results, and also can generate various forms of harassment. And more generally, all this goes into the absurd logic of evaluating people through bibliometric criteria and selecting the stars, as our new CNRS director claims to do, which is, well, everybody who's doing fundamental research know how absurd this is. Okay, so here's for the introduction. So now to the topic. I should also say that I come from mathematical modeling, and I've always tried to stay away from data as much as I could. When I started my PhD, we used to say that data was just a 4-letter word, and now it's becoming really impossible to stay away from data. And this is illustrated, for instance, with the current COVID-19 pandemic. Here I'm just showing some of this data we have from France, because it's an illustration of how it is used in public health. These are the number of detected cases in France over time. And you can see that it's log scale, you get an exponential growth starting in March. And this is the data that's classically used in epidemiology. From this, you can estimate basic metrics, such as the doubling time, the number of days it takes to the epidemic to double in size, or the basic reproduction number, which is the average number of people infected by an infected person throughout his or her infection. But the novelty, and we also see it in the context of the COVID-19, is that it's becoming more and more feasible to collect additional information, especially sequence data. And for the virus that causes COVID-19, the SARS-CoV-2, we've had an unprecedented gathering of such data, with more than 300,000 genomes collected. This is interesting because you can analyze this data using what are called phylogenetics methods. In a nutshell, the idea is that you're going to use a parsimony principle. So you're going to say that viruses viruses come from infections, they come from patients, and you're going to say that viruses that look alike must be closer in the history of the pandemic. And so you can generate a phylogeny, which is like a genealogy, but with rooted in time. And this has been done for decades when it was for species phylogenies, but it is really powerful for infection phylogenies. So here it's just an illustration of how this can be useful in daily public health if you want. On this phylogeny here, the time goes from left to right, and the colors show the regions in France. And you can see many things. You can see some level of clustering between regions, which makes sense. You can also see that there's a big clade here and several additional clades. And so this is kind of an indicator of multiple introductions. So you can see that the first introductions in France here didn't have any progeny, whereas these introductions here led to the massive epidemic wave. And you can also date events. So basically, in a nutshell, sequence data, because here the only thing that is used for this figure are dated sequences, can tell you more about an epidemic, its structure, and even date some important events. There has been a really huge progress in phylodynamics with the use of Bayesian approaches, because the maximum likelihood approaches were facing some kind of a limitation, a wall in terms of inference. And the Bayesian approach allowed to lift some of these limitations. I won't go into details, but basically what you can do nowadays is if you're interested in, well, if you have some data, and with this data, your goal is to estimate an evolutionary model, so how the mutations accumulate into the sequences. But you can also be interested in estimating epidemiological parameters. For instance, these doubling times I mentioned, or this basic reproduction ratio. And finally, you're also interested in this phylogenetic tree to capture the structure. And if you turn the problem into a Bayesian problem using Bayes rules, you can basically break this down into several likelihoods, and some of them are really well known. The phylogenetic likelihood is basically Felsenstein's development in the early 80s. But you can also obtain a likelihood for a transmission model. So the big step forward with these Bayesian phylodynamics or phylogenetics models is that it allowed to be more specific regarding the population dynamics, not just the evolutionary dynamics. There are some challenges, and this is what I'll be talking about today. A first challenge is that in the current models, you need to express and calculate the likelihood of observing your data, of observing your sequence data. And especially if you want to factor in some epidemiological details, expressing this likelihood can be very challenging. The second issue is that the data sets are becoming larger and larger, and these methods, the phylogenetics methods, have been developed with a completely different philosophy. The idea was to extract as much of the signal as you could from a few, like a handful of sequences. So now the problem is now completely reversed. Plus, this data can also be heterogeneous, and I'll come back to this to the end. Another limitation is that you want these approaches to be used in public health. So you have to demonstrate that there's truly an added value to this. If you're just re-estimating things you can estimate with incidence data, it's not really interesting. And I won't be talking about the last point, but it's also a current limitation, which is that these models typically assume that evolution is neutral, and factoring in non-neutral evolution is also a challenge. So in the team, we've been developing methods that can address, I think, some of these current limitations using approximate Bayesian computation. Those who work on HIV epidemiology probably know the ABC, but for another with an other meaning, it was used as a prevention policy. It is also, for statisticians, more known as a method, which is you could consider it a likelihood-free method. In a nutshell, the idea is that you're going to simulate many data sets that have the same structure as your real or target data set. And then you're going to compare these simulated data sets with a target data set to generate a posterior distribution. So you're really trying to look at what the best simulated data, well, which are the simulated data sets that look the most like your true data set. It's particularly appropriate for phylodynamics because there's a very nice parallel between the transmission chain and the phylogeny. Here, time goes from the top to bottom. I'm showing 3 stochastic simulations of a basic epidemiological model, and these are not only the number of infected hosts at a given time. It fluctuates through time. The tree on the right is the exact correspondence of the black curve. And what you can see here intuitively is that there's really a parallel between the two. And in fact, you can go from the tree to the curve directly. All you need is to sum the branches at a given time, and you know the number of infected individuals. And also inside the tree, if you have a full sampling, you can see each branching, each internal node as an infectious event, and each leaf as the end of an infectious period, as a recovery, if you want, or a death. And so it also means that if you run a simulation on an individual based or event based approach, you can very easily generate a transmission tree that can then correspond to phylogeny. So this is what we did, and for this we've developed an R package that should be out very soon called TIPS. The goal was to be as flexible as possible, so it basically works with any ODE system as an input, but we also needed something really fast because, as you'll see, for ABC is a very greedy approach from a computational standpoint, so you need many simulated data sets to make your inference. And currently we were very limited with what was available. So here this is done using basically a C++ implementation in R. And we wanted outputs that could at the same time be time series of phylogenies. And we wanted also to be able to control for the sampling of the phylogeny. So there's a preprint, and yeah, the R package should be out soon. In the interest of time, I'll skip on these aspects, but basically its tips is really accurate and really fast. So trust me on this, and we can come back. Now, I mentioned earlier that with ABC, you want to compare your simulated data set and your real data set. And the difficulty when it comes to phylogenies is that you want to compare trees, and it's a little bit as if you had to compare these 2 trees. There's no single simple metric that will tell you how different these things are. So here, our approach was just to bet on the numbers and to derive as many summary statistics as we could think of. So some of them are really intuitive, like the branch length, their mean length, or the variance. But you can also look at metrics about the imbalance in the tree. I mean, anything you can think of, really. And this is described in a paper from Emma Saulnier's PhD. You can also use the lineage through time curve, which is the number of active lineages at a given time. And same thing, you can get lots of summary statistics from this. What we do then is use regression ABC. So here what we do is that we learn a statistical model based on these summary statistics from the simulated models. So we know which parameters we put in, and we know what summary statistics value come out of the model. This is a representation in 1 dimension, but of course we're in a much larger parametric space. And so you can learn the statistical model, and then when you have a target summary statistics, so for the target tree, you only know what you're looking for, Well, basically, you can then, first with the rejection step, obtain a first distribution, and then with the regression model, reduce the heteroscedasticity and obtain a better posterior distribution. And we do this using machine learning models. These can be linear. We've tried ridge regression, lasso, or now we're working with elastic net, which is a bit of a mixture between the two. You can also use nonlinear models like support vector machines, neural networks, random forest, but personally, I'm a bit more worried about these because of the overfitting problem.
Samuel, can I ask a question about this?
Please do.
So you come up with a whole bunch of summary statistics to then estimate the posteriors from the simulation, right? This is how I understand it. But how do you know that these summary statistics are all sort of like independent, right? It could be that you have, like, to make a sort of very exaggerated, you have a one parameter model, and as you vary this one parameter, 100 statistics co-vary with each other, and now you can measure all these statistics and then calculate the likelihood, but then aren't you going to totally overestimate the sort of likelihood differences?
Yeah, it's an excellent point. So I'm skipping a bit through the details, but we've described it a bit in Emma Saulnier's paper. In a nutshell, what we were really surprised with is that at first we were expecting to select a few summary statistics, like 4 or 5. And what we realized was that the information is spread everywhere. It's very difficult to find summary statistics that have much more information than the average. What we do now is that we also control for this correlation between summary statistics. So we use these variance inflation factors to try and do some selection on the summary statistics. But in general, that's one of the advantages we found with these machine learning algorithms, such as the elastic net, is that they're quite good at handling the multiplicity of summary statistics, and even potentially the fact that they're correlated. And to go to even further back in time, some people have tried to use ABC in phylodynamics before. There's a very nice paper by Oliver Ratman, but he was using the rejection algorithm. And we think this rejection algorithm is much more sensitive to the number of summary statistics. So here, yeah, in 2 steps. First, we do some selection of variables. And second, I also think the machine learning are quite robust to that. So where was I? Did I answer your question?
Yeah, I guess sort of.
Okay.
I mean, it's still quite complicated in my mind, right? Because you have some data set, and then you're not calculating the likelihood of the entire data, but of a subset of some statistics that represent.
Yeah, totally.
And you're estimating those by looking at how often the statistics are something when you simulate it.
Yep.
But you're not trying to simulate the conditional dependencies between all these statistics. You sort of treat them as independent, right?
No, so the model will, you will learn, here I'm only showing a univariate representation, but when you're learning your model parameter, you're learning it on all the summary statistics at the same time.
I understand, but you're treating those as independent in the estimate of the posterior distribution.
Yeah.
Okay, thanks.
Right.
Yeah, it's just very hard for me to see through when that approximation might break down.
Yeah.
Okay, I think we should go on. I mean, I think
I'll
first on what the answer is.
So I'll just show in a few slides an application of this. Just to mention that one advantage is that contrary to the MCMC, you can parallelize your calculation. Okay, so just to show very quickly, we've applied this to an epidemic of hepatitis C virus going on in Lyon. And the originality in this epidemic was that there are 2 types of hosts with 2 different profiles, which I can describe later if you want, but let's call them just the red host and the blue host for now. We had sequence data. This is what the data looks like. You can see there's some clustering, strong clustering of the red of the blue host. And we ran our ABC approach using a two-host models, which is interesting because usually in phylodynamics, it's difficult to have an epidemic with two types of hosts like this. So this is just a basic birth-death model with two host types. It remains a Bayesian approach, so you have to define priors for your parameters, and we had 3 periods of time, basically before the new host appeared and after the new host appeared, as you can see here, blue host and then red hosts. And our goal is to try and estimate these parameters, which are the duration of the infection, the advantage in transmission the new hosts have, and the assortativity of transmission. Do red hosts create red hosts, or do they create blue hosts? And the reproduction number. I'll skip that. As I was saying, we break our phylogenies into summary statistics, with the addition here that the leaves have colors. So we're also dealing with this kind of internal structure. This is what I was just discussing about testing first for, well, removing the variable that might be too correlated to the others, and then performing a rejection, the weighting. And how much time do you have? So we tested did some cross-validation on the method with a leave one out approach. So you just remove one of your data sets, you learn on the rest, and you try to see how well you infer the data, the values you left out. It's working quite well for some parameters, less well for some others. These are how you do with the priors, and this is what you do with your posterior distribution. At least we do better than the prior, I think, for all of the parameters. And these are the estimates you obtain in the end, where in gray you have your prior distribution, and in red the posterior distribution. So it's quite accurate for well, at least yeah, for some of the parameters, some unknowns for others. And this is an additional layer of validation. We did some parametric bootstraps. So we simulated data sets using the posterior distributions, and to see whether our original target was within these simulated data sets. And you can see that when we use the posterior distributions, it works well. If we use the uniform posterior distribution, you kind of remove the information from the posterior distribution, you see that it's less good. And so the summary for the Lyon case is that we find a strong assortativity between host types, and we're also able to detect a difference in epidemic spread with very different doubling times. The new hosts have a much smaller doubling time, so much faster growing epidemic than the classical hosts. Okay, so in conclusion, ABC phylodynamics is interesting because you do not need an analytical expression for the likelihood. It's a highly parallelizable process, contrarily to MCMC, which is much more difficult to parallelize. It can eventually, it is efficient to deal with many summary statistics. And something I didn't present, but it's also useful to perform data integration. For instance, if you want to use the sequence data, but also combine it with classical incidence data. There are several limitations. The choice of the summary statistics, or the definitions of the summary statistics, is really important, especially if you're adding details in your epidemiological models. We've made several assumptions along the way. There's still the phylogenetic uncertainty to deal with, because contrary to the classical Bayesian approach, we're not estimating the phylogeny when we do the inference process. We estimate the phylogeny separately. There are some identifiability issues for some parameters, and it remains still kind of a computationally greedy. And some perspectives, we'd like to investigate non-Markovian effects, contact networks, co-infections, non-neutral evolution, and we've started to look a little bit into model comparison, but yeah, this is still largely ongoing. And with this, I'll thank you again for your attention. Some advertising for Peer Community, which is a peer review initiative. that has been launched recently, and there's now a PCI, a peer community in mathematical and computational biology. So I really encourage you to check it out. And with this, I'll be happy to answer questions if you want.
All right, I'll do the clapping for everybody. Thank you very much. Samuel, questions? Has anybody typed? No, nobody's typed anything yet, but if you have a question, you wanna, otherwise I'll ask a question. I mean, maybe so. I sort of had a similar question to Eric's about how do you actually pick the summary statistics?
Yeah. Right now, as I said, initially we really thought we could find a few informative ones, and this is the way you usually do it in ABC. Here we just took the opposite and basically used all the summary statistics we could think of. One of the things I can show perhaps to give some more. Sorry, I stopped.
But I mean, is there sort of, you know, some sort of multiple hypothesis testing equivalent that you need to do then? Do you need to worry about that?
I'm not sure, because the summary statistics. So what we did was to look at the families of summary statistics and see how they were informative. So you can have summary statistics, for instance, on branch length or on phylogeny topology, on the lineage through time. And then you can look at how well they're correlated with R0 or infection duration or population size. And you can also see how much information they bring. This is a correlation test performed one by one. So you can clearly see that families of summary statistics may bring different kinds of information. I'm not really sure that multiple hypothesis testing is a problem here, because
I mean, I was more thinking about the moral equivalent than actually that, but just if you know, if you basically build statistics that are very similar to each other, that increases your likelihood, increases your score, but you're basically you're giving the same evidence a couple of times. So
I think the worry would be if you lose power by doing this. So if you're just diluting your signal, but since you're then performing cross validation to check on your ability to make predictions.
Okay.
My worry is more this:
Maybe I have
that
a
you're
related
diluting your
question.
signal.
I mean, so what is it essential then that you're really just trying to estimate the entire set of parameters, so that you're not trying to do marginalization of parameters that you don't care about, right? Because when you're starting to sum likelihoods over subsets, then this might become more of an issue, no?
Yeah, this is a good point. You can in the inference try to keep some parameters noisy and don't try to estimate them. I am not sure. We didn't look carefully at how that affects the results. Another potential issue with these approaches, and this is also why I said I'm more worried. I'm less tempted to use nonlinear regressions, is because I can easily see how you would learn your simulation algorithm, for instance. And so something we didn't do also yet is to basically simulate and learn using different algorithms. So you can use a simulation algorithm, say something that would be more detailed on the individual based level, and still use our simulator to run the training data set. Because here I can see how you could, we could be too optimistic about the inference method because we're using the same the same program for both aspects.
So we have a next question from Brent Ullman, precisely about simulated data. Could you explain how the simulated data is generated from the summary statistics? Is it not a resampling of your real data?
So
it's in the chat if you want to read
it. I saw it. So the summary statistics are from the simulated data. So we use this, it's an event-based simulation model. It's really it's based on the Gillespie or variation of the Gillespie algorithm. So you're just simulating a Markov process, and from this you can generate obviously a trajectory, but also if you record the transmission events, you can generate a phylogeny from this, and then the summary statistics are taken from this simulated phylogeny.
Yeah, so next question, Martin Lebert-Eil, is the approximation method similar to the one used by INLA, on which approximation method is used here?
I'm not sure what INLA is, I'm sorry.
Martin, do you want to add something?
Is the integrated nested Laplace approximation?
Okay, I'm sorry, I'm not expert enough to answer.
It's probably not the same one. Yeah.
Evolutionary scenarios based on that. So here, yeah, the evolutionary part is really on the side, and that's a limitation because we work from the phylogeny. So your evolutionary process is already dealt with in a way in when you infer your phylogeny, which is a difference with the classical Bayesian approach. In theory, you could hope to work directly from the sequences using the ABC. But well, the ERC wasn't interested. I submitted that, but in theory, it should be possible to have the evolutionary step nested in the ABC.
Okay, do we have any more questions? I see some people unmuted, so I'm... Anybody else? No?
Yeah, Guillaume, yep. Let me...
Oh, Guillaume, have you compared your inference results with other approaches?
Yeah, so this is, I think should be online. We've compared, that was in the very first study, we compared ourselves to the Bayesian approach with BEAST. And then at the same time, we were comparing lasso and neural networks. Knowing that in BEAST, they have the likelihood of the model. So in theory, you can't really beat it, but they use a numerical approximation, which is why sometimes they are not spot on. The target values in black, you can see that BEAST for R0 is really difficult to beat, and then we were not really far. Infection duration, we're not that far either. And for the population size, you can see that BEAST was doing very weird things for the population size in the SIR model. We're a bit less, how to say, lost, but yeah, so we tried to compare it with BEAST. There's another method by Eric Volz that we should also investigate, but it was a, yeah, we've had issues in implementing it.
Okay, I think I have actually more biological questions, but in the interest of time, we should move on because we're already into the next speaker. So let's thank Samuel again. And